Hello and welcome to the first poster session of the COPAC Symposium. We are going to present 6 posters realized by students from different universities. I will present my work, Quantum Dot Dimers, a Spectroscopic Insight on Interaction Mechanism. I'm Carlo Nazareno Di Benedetto, a PhD student from the University of Bari, Aldo Moro. Next. So my work aims at synthesizing and characterizing dynamic structures in solution based on semiconductor cadmium selenide quantum dots connected by a bifunctional linker and with a well-defined interparticle distance in order to investigate the quantum dot electronic interaction and possible coupling phenomena useful in optoelectronic devices. Next. The dimer formation is promoted by using ditiols as bifunctional linker, thanks to their high chemical affinity with cadmium selenide. However, the high steric hindrance of the native capping layer limits the effective binding. To overcome this issue, a two-step approach is here proposed, consisting in the partial replacement of the pristine ligand with an intermediate ligand, butylamine, having a short alkyl chain and a binding affinity to the quantum dot surface lower than thiols. Such a preliminary treatment is expected to lower the steric hindrance in the organic capping layer, favoring the new ligand binding. The ditiols used have the alkyl chain length that range from nanometer to the sub-nanometer. Here are reported the TM characterization images for heterodimers fabricated with the propan ditiol. Next. The quantum dot proximity in the dimers and the consequent overlap of the wave function may also affect the emission properties. A significant quenching of the photoluminescence in presence of ditiols is observed and gets even stronger when the alkyl chain length reduces till the sub-nanometer distance. Moreover, a redshift in the photoluminescence is observed in presence of ditiols that induce dimerization. Such a difference in the photoluminescence shift cannot be only attributed to an electronic reconfiguration of the energy band after the thiol binding. The decay profiles show a faster decay dependent on the ditiol alkyl chain length, being more rapid for smaller interparticle distance. Next. These differences between the recombination dynamics when varying the alkyl chain length are also reflected in the transfer rates, here plotted as a function of the interparticle distance for both HOMO and heterodimers. At very short distance, the cascade-like arrangement of the electronic levels in the heterodimers occurs in a higher transfer rate. This mechanism can be rationalized with a strong delocalization of the quantum dot wave function that overlap in the space in between the quantum dots. Increasing the interparticle distance, the Fret mechanism starts to be relevant, reflecting in an opposite outcome in the transfer rate, resulting faster in both homodimers as compared to the hetero ones, and depending on the quantum dot size. Next, let me thank the project for this research and all of you for your attention. So the next, let me to introduce. Next, the Matteo Bruschi from the University of Padua with the next poster.
Thank you, Carlo. Good afternoon. I'm Matteo Bruschi from University of Padua, and today I would like to discuss my poster entitled Exciton Dynamics and Optical Response to Stochastic Hamiltonian Evolution. Next. Can you make... Yes, thank you. My research focus is on molecular excitonic systems, which are a system which, due to interchromophoric coupling, have a delocalized electronic excited state. This system have particular interest for photovoltaics as well as for parallel and quantum computing. However, the ability to use this system strongly depends on the ability to control the interaction with an environment, which can lead to decoherence and relaxation phenomena. However, the explicit inclusion of an environmental degrees of freedom is often problematic, so we decided to adopt a stochastic approach. This stochastic approach allows us to include to the system Hamiltonian a stochastic modulation term of the chromophore excitation energies. This stochastic modulation can be assumed to be a stationary Markovian process, and depending to the different correlation time, we were able to distinguish two different limiting cases: the case of static disorder and the case of a fast modulation limit, also known as white noise. Can you make the full screen mode? In the case of exciton dynamics, in presence of white noise, we have that different realization of the stochastic Hamiltonian give rise to very different stochastic evolution, as we see in the picture with all these pale lines, and each of which is ruled by a stochastic Schrödinger equation, an equation of motion which contains a fluctuating term. We also note that decoherence emerges as a consequence of the averaging procedure, as we see from the blue line, which have this damping behavior. And furthermore, in the white noise limit, we were also able to obtain, upon an ensemble average over the stochastic Schrödinger equation, an equation of motion for the density matrix known as Aiken-Strobel-Musser equation, which contain a decoherence term. This term induces very different kind of effects on the exciton dynamics, but especially it gives rise to a reduction of exciton mobility. What we see here is that the slope in the mean square side displacement decreases from be quadratic in time. This is the so-called ballistic regime to a linear regime or diffusive by increasing the decoherence strength. And finally, we probed the linear and nonlinear optical response of the system using 2D fluorescence spectroscopy via a phase modulation scheme. This kind of scheme consists in 4 phase modulated pulses, which are collinear, and the detection of the signal is incoherent using fluorescence. In this way, we were able to resolve the various phase matching directions. What we implemented is a non-perturbative scheme, which for weak intensity, we were just resolving linear and third-order signals, whilst at higher intensity also fifth-order signals can be detected. And finally, I would just mention the effect of decoherence, which caused the disappearance of certain phase matching direction, whilst other are remains. So thank you for your attention, and now I would just like to introduce my friend and colleague Federico Gallina.
Okay, thank you, Matteo, for your kind presentation. I'm Federico Gallina, and I will talk about the equilibration of a quantum walker in presence of disorder. Please, next slide. Next. Okay, thank you. So what is a quantum walk? The quantum walk algorithm is the quantum counterpart of the classical random walk that describes the motion of a walker through the vertices of a graph. In particular, we are interested in the so-called analogical simulation, in which a dedicated device is prepared for the simulation of this specific algorithm. So we can imagine a simulator composed by a lattice, whose sides represent the vertices of a cyclic graph. What happens in a realistic simulator is that these sites can present some irregularities in their energetics that we interpret as disorder in the diagonal terms of the corresponding Hamiltonian. And we have studied the effect of this disorder in the equilibration of the algorithm. Next slide, please. Thank you. So there exist two limiting cases for disorder, the strong disorder case and the weak disorder case. If disorder is strong, we observe Anderson localization, in which the dynamics of the walker is restricted around the initial side of the walker. And this case is not what we are looking for, since this kind of simulators can be used to replicate the algorithm. On the contrary, if disorder is weak, the walker can spread over the lattice. But differently to the ideal algorithm, whose equilibration has just a single relaxation time, now the dynamics present two timescales, a pre-equilibration dynamics that leads to a steady state independent from the disorder, and then an equilibration dynamics that leads to a final distribution that strictly depends on disorder properties. What is interesting is that the steady state distribution corresponds to the target distribution of the ideal algorithm. And we call this a resilience of the algorithm to weak disorder. Please, next slide. Thank you. Okay, now a really brief overview over the main results. So we have studied the spectral properties of the disordered Hamiltonian, finding a spectral gap between two sets of frequencies when disorder is weak. A highest frequency set of frequencies that are in common with the ideal algorithm and are responsible for the pre-equilibration dynamics, while another set of frequencies that is lowest, that is typical to the disordered algorithm Hamiltonian, and is due to a symmetry breaking in the system. And this is responsible for the equilibration dynamics. We have also characterized the lower frequency spectrum analytically, finding a Rayleigh distribution. Then we characterize statistically the equilibration distribution, finding a slightly different distribution than the target one of the algorithm. And what is more interesting, we found an upper bound for the convergence time, also said the mixing time of the algorithm, to the pre-equilibrium. That indicates that the quantum walk on a cycle graph converges faster than the classical random walk for large dimensions. Thank you for your attention, and now let me introduce Pietro De Checchi from Università degli Studi di Padova.
Thank you very much, Enrico. So I'm Pietro De Checchi, currently a fellow researcher at the University of Padova. Next slide, please. So my research is about the external topology in cadmium solenoid clusters, starting from the electronic structure computation to the design of tailored systems to enhance particular design features. The building blocks we are using are clusters belonging to a series composed of stable structures with an increasing number of cadmium atom centers. Short notation, so we have CD4, CD8, CD10, and CD17, and we're focusing on CD8, CD17 for the majority. We systematically investigate the effects of candidate exciton delocalizing ligands. The ligands proposed are characterized by DCOA terminations and a conjugated backbone, and they're namely DTMT and PTNS, while the second one is proposed to add a directionality to the electron density displacement by using a different nitrogen termination at only one of its ends. The effects of the aggregation are then investigated by building dimers using these molecules as covalent bridges. About the computational methodology, all the excited states calculations are performed within the TBFT theory using a range-separated functional and a flexible basis set, reported in the right panel, and chosen after a benchmark analysis. Next slide, please. So to investigate the topology, we recover information from the transition density metrics, moving from a many-body to a two-particle description, so just hole and electron particles. We schematize the system into two fragments and obtain numerical descriptors. The most important are the participation ratio, which tell us in how many fragments each particle is localized, and the charge transfer character, which tell us by what extent the two particles are localized into different fragments. Example of simple limit situation are in the first figure on the left-hand panel. Each state can be further analyzed by the graphical depiction of the spatial distribution of the Holoden electron, the natural transitional orbital, or NTO. This enables to actually see the nature of each state and to observe superposition of configurations. The pair at the bottom of the left panel highlights the feature of maybe the more interesting transition of the spectrum on the right. This spectrum is an example of a modified monomer, and we can clearly distinguish three different zones. The first one is composed of transition of the solely DTMT ligand. A second one, at higher energy, is with the highest contribution of the inorganic cluster. And then we see the rise of a zone in the middle, composed of tens of that transfer. And the most interesting transition are the one at the interface between the second and the third zone, gathering both their features. Looking at the NTO on the left, we clearly see a superposition of a charge transfer state and its directionality, and a delocalized configuration, which gives the oscillator strength and makes this state accessible by optical excitation. Next slide, please. Okay, so an optical excitation doesn't access only one state, but a superposition of those. So to study the actual behavior of a system, we simulate its dynamics upon the excitation with an ultra-fast laser pulse. First, we consider the system after a complete decoherence, enabling to consider the density difference between the ground state and the final superposition in terms of sum of population of the excited state. As a result, we can see that even exciting the system where the states are mostly localized on the inorganic cluster in a dimmer, there is an overall displacement of the electron density towards the bridging ligand. Based on these results, with the first candidate ligand, and by noticing that CD17 monomer is prone to withdraw electron density from the ligand, we model an heterodimer to enhance charge separation upon just optical excitation. And indeed, we can see on the right that we obtain a charge transfer state where there is a displacement of one electron from one cluster to the other at even a great distance. So that's the end for me. Let me introduce you Elisa Fresca from the University of Padova.
Thank you. Good afternoon, everybody. My name is Elisa Fresca, and I'm from University of Padova. Next slide, please. Today, we talk about the role that hydrogen bonds plays in modified electronic properties and the ultrafast dynamics of biological and biomimetic systems. For this purpose, we applied two-dimensional electronic spectroscopy that is a powerful tool since it provides information on the temporal evolution of both coherent and non-coherent processes with a 10 femtosecond resolution. For our work, we've taken in exam and compared a biological pigment protein complex, the water-soluble chlorophyll binding protein, and an artificial H-bonded BODP dimer. As regards the OSCP, it is a tetrameric protein where each subunit binds only one chlorophyll molecules. And we've studied two types of OSCP, one reconstituted only with chlorophyll A molecule and one reconstituted only with chlorophyll B. Therefore, in both complex, the four chlorophylls can be considered as forming two equivalent excitonic dimers, with the four excitonic state expected to be almost 2x2 degenerate. From our 2DES experiments, we have found that in both protein, the ultrafast dynamics follows the relaxation from the higher to the lower excitonic states. But while for chlorophyll A binding protein, the estimated energy gap was in agreement with our calculation, for chlorophyll B, surprisingly, the estimated energy gap was much higher than the calculated one. Since the two proteins are the same, so there are no structural differences, the only difference is the presence of a 4-mil group on the chlorophyll B molecules. So we assume that the establishment of specific and directional H-bond between the protein backbone and the 4-mil group of the chlorophyll B moiety can tune the electronic properties, and so the transitional dipole moments, the excitonic energies, and the exciton gap of chlorophyll B binding protein. Turning to the BODP dimer, we have performed 2DES experiments on the dimer and on the monomer composing the dimer in order to highlight possible differences. And what we found is that in both monomer and the dimers, there is an intramolecular relaxation pathway, which is the relaxation of the vertically excited Frank-Condon state mediated by solvent relaxation. Indeed, in the dimer, we found an additional component that is an intermolecular mechanism, namely a population transfer from the first excited state of one monomer to the first excited state of the other monomer. So we assume that the hydrogen bonds activate new ultrafast dynamics channels in the relaxation of H-bonded body P dimer, both with an intra and intermolecular mechanism. These findings were also supported by our beating analysis, and what we found is that only in the dimer there are beating modes that are attributed to vibrational modes of functional group implicated in the formation of hydrogen bonds. So in conclusion, all these findings suggest that the presence and establishment of H-bond is a powerful tool to drive and tune the photophysical property complex system by specifically engineering the interaction with the surroundings. And now let me introduce you my colleague Nicola Peruffo from University of Padua.
Thank you very much, Elisa. So good morning, everybody. Nicola Peruffo. I come from University of Padua. I am a PhD student. Next slide, please. So today I'm going to present you my work, that is the selective switching of multiple plexitons in colloidal materials, directing the energy flow at the nanoscale. Next slide, please. First of all, we have to understand what is a plexiton resonance, which indeed is a light matter hybrid resonances. And yes, light, first of all, is entrapped onto plasmonic materials, such as nanoparticles or surfaces, while the matter counterpart could be done by, for example, molecules, aggregates of molecules, or either quantum dots. And they are disposed on the surface of the plasmonic entity. Well, when the plasmon mode and the excitonic mode couple together, new plexitonic resonances occur. And, as you can see from the image, they are an upper and a lower resonance, and they can be also be spotted in the Stixio spectrum as a new band. These materials are really interesting for nanophotonics and quantum technologies. Next slide, please. So here my material is made by gold nanoparticles, functionalized with cationic alkyl thiol and an anionic porphyrin, which can be tuned into aggregates by selecting which kind of environment used. So next slide. As you can see, we can tune the aggregation form of the porphyrin by selecting the ratio between porphyrin and nanoparticle. With the low ratio, we have just the porphyrin in monomeric form, and we have the interaction between the porphyrin and the nanoparticles. So, looking at the right image, the Q-band of the monomeric form of the porphyrin disappear, and two new bands appear. These are plexitonic bands, and they are called Q-plexiton. At high ratio, instead, the porphyrins aggregates onto the surface of the particles into J aggregates, and these aggregates interact with the plasmon mode of the particle. So two new resonances appear instead of the V band of the J aggregate form of the porphyrin. And as you can see from the right image, they are, for example, called upper and lower resonances of the B plexiton. At intermediate regime, both the resonances are switched on, and also we are able to find that these two resonances interact each other. So next slide. We excited the samples at the V-band region, and we checked the luminescence of the Q plexiton region. And as you can see, when the sample has both the plexitons switched on, the luminescence is really different from the typical fluorescence of the porphyrin, and is the typical luminescence of the Q-plexiton. So, exciting the B-plexiton, the Q-plexiton emits. And this is the clear sign that these two plexitons are linked together. In conclusion, next slide, please. We prepared a system in which we are able to switch on and off different kind of plexiton. So it is a responsive system in which we are able to make a connection between these two kind of plexitons. So we are able also to direct the energy at the nanoscale. And if I got your attention, please check our new work about it. And finally, I would like to thank my group, the MUOS group, and my supervisors, Elisabetta Collini and Fabrizio Mancini, for their helpful advice. Finally, I would like to thank also University of Padua. Thank you. So, my presentation has come to an end. We now can attend to the conference of Professor Engel, which name is Quantum Dynamics for Real-Time Processing of Excitonic Energy, stunning vibronic coupling to steer energy transfer within a photosynthetic complex. You just have to click on the link present in the chat. Thank you.